So, in the process of photosynthesis, the very first thing that is going to take place is the process of harvesting of the light. And that harvesting of the light, it occurs, or it actually is conducted in the uh, granum region, or the thylakoids of the chloroplast. And this particular process, it goes by the name of light reaction, or to be very specific, it is called light dependent reactions. This light dependent reactions, it is called so because here light will be utilized for the entire purpose of this, uh, like whatever steps that are supposed to happen right there, that does take place actually, for every single of that process to to happen properly. There is an involvement of light there, and without that this entire step is completely impossible. That's why, being dependent on the source of light, this particular process has been held this particular name of light reaction. But it is that this process of light reaction that is going to take place, it releases material, like you can see it's mentioned here. It produces ATP and NADPH, which is then transferred to the like stroma region of the chloroplastid, where by the Kelvin cycle it actually gets converted into the sugar, because sugar is produced by using the CO2 from the atmosphere, and the CO2 getting converted to the sugar, it actually required the assistance of these two things. Thus, the second process, this entire process of production of the production of the carbohydrates from the carbon dioxide by the CO2 fixation step, is what it goes by the name of dark reaction, because it is called so just as a simple moniker, you can say, but the actual name of it is light independent reactions. Now, for this entire like two things to happen properly, the entirety of the process of this light reaction and the dark reaction, they all can be like easily explained with the biochemical steps that are actually taking place within. But to proceed with the entire process, to even initiate the process of this photosynthesis, the very first thing is that harvesting of the light. That the light that is being coming there from any of the source, that it is harvested by the granum. This particular step is the very first step of the photosynthesis. And for that, this particular system is there present within the thylakoid membrane. Yes, it is actually a representation of the thylakoid membrane, on top of which you can find this array, this large array of these different photopigments that are present there. Each and every of the disc that is represented here, each and every of those discs, they are all photopigments, and those photopigments are arranged in this particular array, where we can see that the light is coming. Light is represented as a photon right here, a unit of light, you can say. So this light is actually coming and falling there. So right there, after falling, what is happening there is a resonance transfer taking place. Resonance transfer is nothing but the energy that is uh, or you can say the excitation that this particular pigment, photopigment, it receives by receiving the sunlight. That excitation is being passed to the next to the adjacent photopigment, and then to the adjacent photopigment. This process is necessary for one particular reason, that is that this entire light harvesting complex, it actually, its purpose is to initiate the process of photosynthesis. Yes, and to initiate that for purpose of photosynthesis, there are additional steps that are required, and for that additional step to actually take place, there are systems that are set. There are proteins, there are carriers, there are many kind of molecules that are set one after another, and those things need to work in a in an adjacent transduction process, you can say. And that particular process to happen, it cannot happen with a random places from any other section, you can say. It's not possible from any of the sections. So it has to start from one particular point only, and that particular point is what is known as the reaction center. So you will have one reaction center surrounded by this all this photopigment who are acting as an antenna which will receive the signal and transmit it directly to it, and that is what we are seeing here. The light is coming as a form of the signal here. After receiving the signal, that signal is then passed, that energy is passed by one after the next to the like different photopigments, and finally to the reaction center chlorophyll. And then this transfer of the electron takes place, which is the one that is necessary to trigger the entire the cascade steps of the photosynthesis process, the light reaction. So there you can see that there are different type of photopigments are there, and then there is one photopigment responsible for the entire process of the light reaction, and this entire setup is what is known as the light harvesting complex, because it is the one that is harvesting the light, and that central molecule is the one that's initiate the entire reaction, so it's the reaction center. So if we can express it in terms of like a simple schematic diagram, then we will find that it's like one after another, like all the photopigments are arranged, and then finally the main photopigment is there where the process of the electron transfer is taking place. ETS represented here is electron transport system, which is that cascade of transferring proteins that are there arranged along with this molecule, so that the process of light reaction can take place. That is actually the main process of the light reaction, we can say. And the antenna pigment complex that we are seeing right here, not necessarily that every single type of leaf will have all of them present there, but a multiple of them is definitely present. But we can see one thing very clearly here, that each and every one of them are named differently, as they are a different type of molecule, but always the one that falls in the reaction center, or that can be found in the reaction center in most of the plant or nearly all of the plant, is actually chlorophyll type A. Now, chlorophyll type A can be of two varieties depending on the absorbance range, called the P680 and P700. But the rest of them, they are there just to accept the light energy and then pass it down. So there is chlorophyll A, there is chlorophyll B on top, you can see. Then you have this different other color molecules, the lutein, the zeaxanthin, or actually xanthophyll. Then beta carotene is actually also known as carotene itself. Then we have lycopene. All of them are different kind of photopigment. Each and every of those photopigment have its own distinct color. You can see that the chlorophyll B is quite dark blue type, dark bluish green type color, whereas the chlorophyll A is light green in color. Let me show you a different picture here. This is actually a way of extracting the photopigments. It is like by crushing the leaves and then running it with the help of, like, dissolving it with a proper solvent. We can actually separate them in a separation funnel like this, and you can see that there are these two distinct color that is formed right there, where there is this chlorophyll A in one corner and then the chlorophyll B in the other hand. And this is actually a color chart, we can say. This is a chromatography paper, like in paper chromatography, the entire process is being carried out, and we can see that a mixture of photopigments were used. Well, this is achieved by simply mixing like 2 3 different types of leaves and crushing them together, and then going for the process of this extraction, because a single leaf doesn't have all of those all this type of leaf. Like there are only few, like many, most of the leaves, they don't have all of them present there. But yes, the one that is usually found is chlorophyll A, chlorophyll B, and some extent of the xanthophylls. The phytoplankton and the beta carotene may be present, but that depend on the variety of the plant we are talking about. If like most of the plant, they don't have those material, they are present there. So you can see the different color patterns. There are different types. There are neoxanthins, phytoxanthins. There are different types of xanthophylls are there. Then we have this chlorophyll B, which is like, you can see, like it's kind of um, a dull green type color, whereas the chlorophyll A is quite vibrant. So this kind of way the all the colors are can be separated. And finally this all this color pigments, their purpose, as it has been shown in the previous slide, that its purpose is simply to act as an antenna and to supply the energy to the chlorophyll A, which is the main reaction center. So chlorophyll A is the common thing where every single plant it has the chlorophyll A. The other type of pigments, maybe one or maybe many of them are present along with it as a combination to work as a light harvesting complex. Now, as the different proteins, as I mean, so as the different type of photopigments as they go, their main purpose is absorbance of light, isn't it? So this actually graphical plotation is to show the different photopigments and their absorbance. It's a color coded graph where you can see that each and every of the like photopigments have been represented with a different color, and then they are shown in the form of like what particular range of light they are absorbing. This is the entire range of the visible light that we know, starting from like you have the violet to the red range. The entire range is given there, and the wavelength of the light they majorly absorb, that particular wavelength series of the light is also being you can see it. Like for instance, the pheophyte, the phycocyanin in the center, you can see this blue pigment phycocyanin in the center. It absorbs this particular range of this green to a little bit of orange. uh, like green to orange, this entire range is what is absorbed by the phycocyanin, but it doesn't absorb any other range beyond that. Cannot absorb the red one also, can definitely being a blue color pigment, it will not absorb the blue light. So similarly, you have the carotenoids which absorb the entire blue range and the green range, but it cannot absorb the yellow to red because it itself is a orangish color pigment, so it cannot absorb that particular color range. See, it's always the case that any pigment that we see, it has a like pigments are usually colored, isn't it? But that color is like the color which it cannot absorb from the entire visible range. Uh, Means if it is red in color, then it cannot absorb red, but it can absorb the rest other color, and can only like let the red color come out of it. So that way, like they appear red in color. So likewise, every single photopigment it acts like the same way. But when we talk about the chlorophyll A and the chlorophyll B, its absorbance are slightly different. Its absorbance, like definitely being a green color pigment, it will not absorb the green wavelength light, isn't it? The entire green section it is not absorbed right there, as you can see. But this molecule is capable of absorbing, you can see the entire like the violet to blue range, and also the yellow to orange range, the red even. So this absorbance, this different type of absorbance that we are seeing here, is what makes them the most suitable one to absorb the light energy, because blue light is available a lot, it is absorbing that, and the high energy light is a red range, and which is absorbing that particular range also. So that means the red and the blue, both the sections are being absorbed right there, and depending on that particular point, we can say that becomes the most effective one to go for the process of photosynthesis, because it absorbs the maximum wavelength of the lights. And thus this two molecule can majorly be found as the main photopigment in most of the organism. Means whichever organism is carrying out the process of photosynthesis, you are going to find that the chlorophyll pigment is present there, of which chlorophyll A acting as the most like effective one, being as you can see it has actually the most effective range. You can find the chlorophyll B is absorbing blue light the maximum right there, isn't it? It has a very high peak right there. Okay, means 80% of the light is getting absorbed right there. But it doesn't matter with comparison to that of the red, because the chlorophyll A, because chlorophyll A can absorb a bit wider range, isn't it? It can absorb the orange and the red quite well than that of the chlorophyll B. That's why the chlorophyll A actually is the most accepted light harvesting complex, you can say the most accepted light harvesting complex, the one which is found in the reaction center even. So ultimately, to conclude, like definitely this process is like in the chloroplast, as you can know, that it is the one chlorophyll A which is the one acting as the light harvesting complex. Here the molecule that is shown here is actually chlorophyll A only, because the actual distinction happens right there in this particular section, where if it is a CH3 being present, there is a chlorophyll A, and if it is CHO present there, it is chlorophyll B. Don't fret, this is not going to be asked in the examination by showing you the diagram, and you don't need to remember the diagram as it is. So this picture is just like for reference, you can say. So this is a porphyrin ring, the one that is the main chlorophyll molecule, and it is present right in the thylakoid and is the one acting as the main light harvesting complex and the main reaction center there. Now, the very first step of that process, I'm going to discuss that also, because to trigger the entire process, as I said, it is the one which is acting as a reaction center. But what is that reaction? What does it do? So it actually getting excited means by gaining that light energy, that gaining that photon, it actually gets excited, and that excited